Hi everyone, I'm Dr. Xin from Center for Reproductive Medicine, Shandong University. Today, I would like to share some genetic progress in premature ovarian insufficiency. First, I want to talk about this disease, premature ovarian insufficiency. We often operate it as POI. POI is a clinical syndrome defined by loss of ovarian activity before the age of 40. The prevalence of POI is approximately 1 to 2%. The diagnosis of POI is based on the presence of menstrual disturbance and biochemical confirmation. Although proper diagnostic accuracy in POI is lacking, the following diagnostic criteria are recommended now. Oligoamenorrhea for at least 4 months and an elevated FSH level exceeding 25 on 2 occasions. The sequelae of POI include loss of infertility, reduced bone mineral density, increased risk of cardiovascular disease, etc., and even it will reduce the women's life expectancy. Here is a change of oocyte number during female lifespan. At the 20th week of gestation, there are 6 to 7 million germ cells, but later, atresia dominates and its influence, only 1 to 2 million germ cells remain at the time of birth. In the beginning of menarche, the number of approximately 500,000, and later, depletion occurs at a constant rate of around 1,000 per month. Under normal conditions, only about 400 follicles reach the mature pre-ovulatory stage. POI can result from a small pool of primordial follicles or premature follicles depletion due to accelerated atresia. The disorder is highly heterogeneous in etiology. A wide spectrum of causes have been considered, including genetic, autoimmune, infectious, or iatrogenic. Respectively, the majority of patients remains to be unclear about their etiology. Genetic causes account for approximately 20 to 25% of the patients. Chromosome abnormalities could explain 10 to 50% of POI cases. Yet, identifying precise curative genes for POI has been challenging. Over the past few decades, numerous candidate genes emerged, but few were incorporated as curative with functional validation. Very recent approaches using next-generation sequencing, that is NGS, especially whole exome sequencing in large POI pedigrees, have led to new causatives being identified and candidates being proposed. Here, we review the currently progress in POI genetics. Moreover, we will discuss the challenge and perspective in POI pathogenicity. First of all, I will introduce 2 common approaches used in POI genetic studies, that is, candidate gene sequence and whole exome sequence, and I will focus on WES studies in the latest non-synonymic POI. Here is the 13 genes identified by candidate gene approach. These include genes involved in primordial germ cell migration and proliferation, such as NANO3, or in cell death, oocyte-specific transcription factors, other transcriptional factors affecting follicular genesis. Also, it includes transforming growth factor superfamily, including BMP15 and GDF9, and HOMO and their receptors, such as FSH receptor, AMH, and AMH receptor 2. From these findings, we can see candidate gene approach is based on the gene expression or non-function in follicular genesis or ovarian development, or the phenotypes of murine KO models, However, inconsistent with the homozygous pathogenicity in mice, most of the mutations are heterozygous in human. Additionally, no gene is implicated in more than 5% of the cases, except for a few genes, including BMP15, FMR1, and NOBOX. It falls short of identifying novel or less well-known genes based on priori hypotheses. Usually, the family relatives are not involved in the analysis. Continued advance of NGS will overcome these drawbacks. Although progress slowly, the candidate gene approach has indeed contributed fruitful genetic variants and established the fundamental field of POI, curative genes, and its genetic agriculture. Here is the 33 curative genes identified through WES in POI families. Primary amenorrhea, I operated as PA, means patients present with primary amenorrhea, and SA stands for secondary amenorrhea. Different from genes in sporadic patients by candidate gene approach, 80% of mutations were homozygous or compound heterozygous in these families, which means they were inherited in recessive pattern. 30% were found in consanguineous family. Most of these genes are new for ovarian function, including the purple genes important for DNA damage repair, homozygous recombination, and meiosis. Blue ones responsible for mRNA transcription and translation, and the green ones POI candidates related with follicular genesis. WES approaches in consanguineous and large POI pedigrees have been very promising in identifying known genetic variants. Interestingly, it showed an overwhelming enrichment of genes involved in DNA damage repair, HR, and meiosis. Accumulated DNA damage due to repair deficiency within oocytes could induce meiotic arrest and promote apoptosis, result in accelerated oocyte loss, thus causing ovarian failure. Most mutations are inherited in autosomal recessive fashion in consanguineous families with primary amenorrhea, suggesting their deleterious effect on the underlying germ cell pool. Pathogenic heterozygous mutations only identified in patients with secondary amenorrhea. Of note, mutations in most genes were in biallele state. That means homozygous or compound heterozygous, and the heterozygous carriers exhibited normal fertility, which questions the causality of heterozygous variants identified before in sporadic POR. Homozygous loss of function mutations found in no genes such as FS receptor, SOLO1, and GTF9. Heterozygous variants of these genes have been reported in sporadic cases, which are likely not the cause, or there are other variants in other genes that may interact. Further functional validation and multiple family members are warranted to further decipher their true pathogenicity in sporadic POR. Next is about the NGS in sporadic POR. To date, there are 5 studies using NGS have been performed in sporadic women with POR to propose new putative candidates. Targeted gene sequence focused on no genes as panels of specially selected regions. 4 genes mutations were confirmed with deleterious effect, that is Notch2, ATG7, ATG9A, and C4. Limited sample size and most mutations lacking functional confirmation. Whether the cumulative effect of different genes or different variants supports the polygenicity of POR remains to be elucidated. How to separate the weight of causative alterations from the chef of the vast amount of noise variants is challenging. Now, we make a summary of curative genes of POI. This is the representation of follicular genesis and oogenesis. We labeled the genes according to their functions at different stages of follicular development, from PGC to graft follicle, including mitosis and meiosis process. Here is an overview of POI curative genes clustered during the damage repair, homologous recombination, and meiosis. These genes in blue box were from candidate gene approach, whereas in red box from NGS approach. While stars identified curative genes mostly involved in DNA damage repair, HR, and meiosis. Candidate gene approach clustered the curative genes responsible for follicular development. Above mentioned is the summary of the genes responsible for POR. Next part, I will introduce some perspective and challenge in this field. Thank you.